Hello everyone, welcome to another video of general pathology. In this video, we are going to be talking about cell injury. This one is actually a really important topic because almost everything else you will study in pathology, from heart attacks, liver diseases, strokes, all it starts right here at the level of a single cell getting injured. So if you understand this properly, a lot of pathology later is going to make a lot more sense. So let's dive deep into it. What is cell injury? For us to understand the concept of cell injury, I would like us to look at the bigger picture. That is, when a normal cell is being exposed to either physiological or pathological stress, the normal cell is going to try to find ways in which it is going to survive that stress. So the process in which the normal cell finds ways to survive a particular stress, which can either be physiological or pathological, is what we call cell adaptation, which we have discussed in our previous videos. But when the stress keeps on persisting on that cell that is trying to adapt, the cell that is undergoing adaptation will lose its ability to adapt to that particular stress. The cell now is going to undergo what we call injury. So continuous persistence of stress on a cell that is undergoing adaptation will make the cell to lose its capacity to adapt to that stress, and hence the cell is going to undergo injury. So that is the concept of cell injury. That is why we say cell injury is a functional and structural alteration in a cell produced when an internal or external stress exceeds the cell's capacity to adapt. So what are the factors that determine whether a cell is going to adapt, the cell is going to undergo injury, or we can say the cell is going to die? There are three main things that determine whether a cell is going to undergo those three main processes, and those three main factors are the type of the stress, the duration of the stress, and also the severity of the stress. These three factors are what determine whether a cell is going to undergo adaptation, or the cell can no longer undergo adaptation but undergo cell injury, or the cell is going to die. But there is something worth remembering, that the same stimulus can cause adaptation in one cell type and death in another. For example, if you have two organs that lose the same amount of blood at the same time, one organ can be okay with it and the other organ might die. So the same stimulus can cause adaptation in one cell type and death in another. So the outcomes also depend on the cell's own adaptability status and genetic makeup. Those two organs, their adaptability status and their genetic makeups are different, hence the ways in which they respond to various stresses are also different, even though the stimulus affecting both organs is the same. Now let's look at the causes of cell injury. One of the main causes of cell injury is hypoxia, which is the commonest cause of cell injury, which is the loss of oxygen delivery to a cell. The other one is physical agents, can be trauma, heat, cold, radiation or electric shock, infectious agents and immunologic reactions, chemicals and drugs, genetic derangements such as enzyme defects, misfolded proteins, nutritional imbalances such as inadequate protein or vitamins, or excessive production of cholesterol are some of the causes of cell injury. So just to double down on what I was talking about in the previous video as to what determines whether a cell is going to undergo adaptation, the cell is going to undergo cell death, or the cell is going to undergo cell injury. So there are principles that are guiding it, and some of those factors, as we've mentioned, are the type of injurious agent that is being used, or the severity of the stress, the duration of the stress, the type of metabolic state of the stress, the cell's adaptive capacity, and the tissue and organ context. So these are the factors that determine whether a cell is going to undergo adaptation, cell injury, or cell death. But there is something worth mentioning, that if the stimulus is mild or removed early, the cell can fully recover. If it is moderate but persists, injury is reversible. If it is severe or prolonged, injury becomes irreversible and the cell will undergo cell death. So these factors are very high yield for your exams, and the next thing we'll be talking about is the outcomes. What are the four possible outcomes of a cell that is being exposed to stress? The first outcome is recovery, that is complete return to normal structure. The second one is adaptation. The cell will find ways in which it is going to survive. The third one is reversible injury. When the cell can no longer survive, the cell is going to undergo injury, but that injury is reversible. And the fourth one is irreversible injury, where the cell can no longer return to its original form or to its normal state even though the stress is being removed. So damage passes the point of no return and the cell dies by either necrosis or apoptosis, which we are going to discuss in our next video. So we are going to talk about mechanisms of cell injury just to have a brief picture of it before we start each and every one of them. So the first one we'll be talking about is ATP depletion. ATP depletion is one of the commonest ways in which a cell undergoes an injury. The other one is mitochondrial dysfunction, defects in membrane permeability, and oxidative stress. These are some of the ways in which cells undergo cell injury. So now let's start with ATP depletion. How ATP depletion causes cell injury. But before talking about how ATP depletion causes cell injury, let's look at what are the causes of ATP depletion. How do we have ATP depletion inside our cells? So the causes of ATP depletions are, one of them is hypoxia, mitochondrial damage, toxins and chemicals, ischemia, severe infections, and physical agents. Some of these causes are the reason why we have ATP being depleted inside our cells. Let's say, for example, we have ischemia. Ischemia is reduced supply of blood to an organ or a cell. So let's say an organ is supposed to receive 100 ml of blood, but the organ is receiving 20 ml of blood. The organ is said to be ischemic. So if an organ is receiving low amount of blood, so definitely the organ is also going to receive low amount of oxygen, because oxygen is being transported by the blood. So if there is low amount of oxygen being transported to an organ, then therefore the organ is also going to receive low amount of ATP, because oxygen is necessary for ATP production by the electron transport chain and oxidative phosphorylation. So if there is low amount of ATP going to an organ or to the cells of an organ, the following scenarios are going to happen. The first thing is being mentioned here in the diagram. The first one is failure of NA, sodium potassium pump. So this sodium potassium ATPase pump are pumps that work perfectly in the presence of ATP. What is the job of this pump under physiological condition? The role of this pump is to pump out 3 ions of sodium out of the cell and take in 2 ions of potassium. So meaning they remove sodium ions extracellularly and take in potassium ions intracellular. That is the role of this pump under physiological condition. But if there is no ATP, the pumps will not be able to take out the 3 atoms of sodium and take in the 2 atoms of potassium. Hence, there will be high accumulation of sodium inside the cell. And if there is high accumulation of sodium inside the cell, wherever sodium is, sodiums like to attract water. So there will be high amount of water inside the cell. Hence, that will lead to cell swelling. That will lead to cell swelling and the formation of blebs. These are some of those elevations that you see on the surface of the cell. We call them blebs. So this is how falling of ATP causes cell swelling. Another result of falling of ATP is increase in aerobic glycolysis. So if there is low amount of oxygen going into a cell which causes low amount of ATP, the cell will not undergo aerobic glycolysis. Rather, the cell will undergo anaerobic glycolysis. And anaerobic glycolysis leads to the production of lactate. So more lactate is going to be produced inside the cell, which will lead to the accumulation of lactic acid inside the cell's cytoplasm. So the accumulation of lactic acid inside the cell's cytoplasm will cause what we call lactic acidosis. So accumulation of lactate or lactic acid will cause lactic acidosis, and hence thereby reducing the pH of the intracellular content of the cell. So the second cause of falling of ATP is it leads to the decrease in the intracellular pH by increasing the level of lactic acid accumulation inside the cell. The third cause of ATP depletion is failure of sodium pump. Inside the cell, there is low amount of sodium physiologically, and sodium is found extracellularly in physiological state. But if there is a failure of ATP, or if there is falling of ATP, the cell's calcium level will increase, because there will be nothing holding the cell or the calcium that is found inside the cell outside the surface of the cell. So there will be high amount of calcium content trickling inside the cell, thereby causing the cell to have high level of calcium, which is not supposed to be present inside the surface of the cell. And the last but not least is the detachment of ribosomes. These ribosomes, under physiological condition, the ribosomes are responsible for protein synthesis when they are attached to the endoplasmic reticulum. So for the ribosomes to produce, or for the ribosomes to do what we call protein synthesis, the ribosomes must be attached to the endoplasmic reticulum, and that can only be attached if there is ATP. So if there is no ATP, the ribosomes will not be able to attach themselves to the endoplasmic reticulum, and hence they will not be able to participate in protein synthesis, and hence we will not have proteins in our cell, or we will not have adequate amount of protein in our cell. So these are the four main causes of falling of ATP to the cell. The first one is failure of the sodium potassium ATP, which will cause cell swelling. Increase in aerobic glycolysis, which will increase the level of lactic acid accumulation inside the cell, which will cause lactic acidosis. And the third one is failure of this potassium pump, which will cause high amount of potassium, failure of calcium pump, which will lead to high accumulation of calcium inside the cell. And the last one is detachment of ribosomes, which will prevent the cell from participating in protein synthesis. So these are the reasons, or these are the ways in which ATP depletion causes cell injury. So just to double down on what I was saying, ATP is required for every synthetic and generative process in the cell, such as hypoxia, toxins, which shut down its production. Failing ATP causes failure of the sodium potassium pump, that is, sodium and water influx will happen, and then cell swelling is eventually going to take place. It also triggers increased aerobic glycolysis, depleting glycogen and generating lactic acid, which lowers intracellular pH, just as we've discussed. And falling ATP further causes failure of the calcium pump and detachment of ribosomes, halting protein synthesis. So these are the ways in which ATP depletion causes cell injury. Mitochondrial dysfunction. How does mitochondrial damage causes cell injury? But for us to understand how does the damage of the mitochondria causes cell injury, we have to understand what is the role of the mitochondria when it is not damaged. What is the function of the mitochondria under physiological conditions? We know the mitochondria, it is the site for ATP production through the electron transport chain and oxidative phosphorylation. So if the mitochondria is performing its job properly, it's going to lead to the production of ATP. But in this scenario, the mitochondria is damaged. What are the consequences, or what are the effects of the mitochondria being damaged? So let's look at what we have here. If the mitochondria is performing its job properly, if it is not damaged, it is going to lead to the production of ATP. But if the mitochondria is not performing, or is not performing its job properly, meaning it is damaged, the mitochondria is not going to lead to the production of ATP, but the mitochondria is going to produce what we call free radicals through abnormal oxidative phosphorylation. So when it is performing its job properly under physiological conditions, it uses normal oxidative phosphorylation to produce ATP. But if the mitochondria is damaged, it is going to perform abnormal oxidative phosphorylation. And abnormal oxidative phosphorylation leads to the production of free radicals. And these free radicals, if they are produced, they lead to the damage of organelles that are found inside the cell. So abnormal oxidative phosphorylation leads to the production of free radicals, and the free radicals damages the organelles that are found inside the cell. Another effect of mitochondrial damage is inside the mitochondria under physiological condition, we have what we call cytochrome C. And that cytochrome C only comes outside of the mitochondria during the period of programmed cell death. And that programmed cell death is called apoptosis, which we are going to discuss in our next video. So if there is no apoptosis, the cytochrome C should remain inside the mitochondria, because anytime the cytochrome C comes out, the cytochrome C leads to cell death, and that cell death is called the programmed cell death, which is called apoptosis. But if the mitochondria is damaged, whether there is programmed cell death or not, the cytochrome C is going to come out. And if the cytochrome C comes out, the cytochrome C will lead to programmed cell death called apoptosis, even though it is not needed. And that programmed cell death will lead obviously to the cell being dead. So that is another effect of mitochondrial damage, causing the cytochrome C to come out of the mitochondria, causing the cell to die. Another effect of mitochondrial damage is loss of calcium homeostasis. 99% of the calcium that is found inside the cell is found extracellular. Only 1% is found intracellularly. So most of the calcium is found extracellularly, and 1% of the calcium is found intracellularly, that is inside the cell. And the calcium that is found inside the cell is found in the mitochondria and endoplasmic reticulum. So if there is mitochondrial damage, that calcium that was found inside the mitochondria will come out, will leak out into the cytoplasm. And when calcium is found inside the cytoplasm, it will cause the production or the activation of certain enzymes. The enzymes that it is going to activate are ATPases, endonucleases, phospholipases, and proteases. And ATPases, what do? They lead to the degradation of ATP, leading to ATP loss. Endonucleases, they lead to the degradation of DNA. Phospholipases to the degradation of phospholipids, and proteases leads to the degradation of proteins. So if all these substances are being degraded, what is going to happen is cell injury or cell death. So these are the consequences or the effects of mitochondrial damage on the cell. So let's have a summary here. So we know that mitochondrial damage prevents normal oxidative phosphorylation. And what is going to happen? Abnormal oxidative phosphorylation is going to happen, which leads to the production of free radicals, which damages the cell organelles. Also, cytochrome C leaks into the cytoplasm, which also activates caspases, and apoptosis is going to take place. Caspases are just factors that help in the process of apoptosis. There are different types of caspases. We have caspase 9, caspase 8, which we are going to discuss when we will talk about apoptosis in our next video. Also, we said that loss of calcium homeostasis activates ATPases, endonucleases, phospholipases, and proteases. And these enzymes, what do? ATPases causes ATP degradation, endonucleases causes DNA degradation, phospholipases causes phospholipid degradation, and proteases causes protein degradation. And if all these things happen inside the cell, what is going to happen is called cell injury. And if it is happening in a very severe case, it can lead to cell death. So knowing how mitochondrial damage causes cell injury is very high yield for most of the university exams. So now let's talk about how defects in membrane permeability causes cell injury. So inside our cells, we have certain structures that have membranes. Example is the mitochondrial membrane, the lysosomal membrane, and maybe the plasma membrane. So we already spoke about the mitochondrial membrane. Now let's talk about the other membranes, such as the plasma membrane or the lysosomal membrane. So if there is a defect in this membrane, such as the plasma membrane, certain things are going to happen. So let's look at this picture here. So the first one we have is called the plasma membrane damage. Plasma membrane damage can be caused by disruption of the lipid layer, can be caused by protein damage, can be caused by cytoskeletal injury or toxins such as free radicals and other factors. So some of these things can cause the damage to the plasma membrane. And if there is damage to the plasma membrane, the consequences or the effects are as follows: loss of osmotic pressure, efflux of fluids and ions. So since this plasma membrane is the certain fluids or ions that are found inside the cell are going to go out of the cell. And the third one is loss of cellular content. Some of the contents that are found inside the cell are going to be released out of the cell, hence going inside the extracellular space. And the last but not least is called cellular swelling and lysis. Since there is plasma membrane damage, the fluids that are also found outside the cell, extracellular, are also going to enter inside the cell, such as water. Water is going outside the cell and then enter inside the cell, causing the cell to swell. And all these effects will lead to cell injury. And if these effects continue to persist, they might trickle down to what we call cell death. So these are some of the ways in which plasma membrane damage can cause cell injury. The other type of membrane we have is called the lysosomal membrane. And inside this lysosomal membrane, we have what we call certain substances that are found inside the lysosomes. We have hydrolytic enzymes that are found inside the lysosomes, such as proteases, nucleases, phospholipases, and glucosidases. Some of these substances are found inside the lysosome. So if there is damage to the lysosome, and the causes of that damage might be a rupture, membrane destabilization, or even toxins, can cause lysosomal membrane to be damaged. And if there is damage to the lysosomal membrane, the effects are as follows. So if some of these hydrolytic enzymes are exposed to the cytoplasm of the cell, they can cause damage of the DNA, they can cause damage to the RNA, and also they can cause damage to other cell components, which can lead to cell injury. And if that damage continues to persist for a long period of time, it can lead to cell death. So these are the ways in which defects in membrane permeability can cause cell injury. Now let's talk about oxidative stress and free radical injury, how it leads to cell injury. So for us to understand free radical injury, or how oxidative stress leads to free radical injury, we have to know what is a free radical. So what do we say is a free radical? A free radical is an unstable chemical species with a single unpaired electron in its outer orbit. So an unstable chemical species with a single unpaired electron in its outer orbit. Free radicals arise from either increased production or ineffective degradation. So if there is increased production or ineffective degradation of substances, free radicals are being produced. So now, what are the conditions? What are ways in which free radicals, or what are the conditions that favor the production of free radicals? Free radicals are produced extensively in the process of inflammation. When inflammation happens, we know that inflammation is a response to the connective tissue injury. Anytime we have a connective tissue injury, the body responds to the process of inflammation. We are going to talk about inflammation in our next videos. Another factor that promotes the production of free radicals is chemical or drug injury. If there is chemical or drug injury, free radicals are going to produce radiation and ischemia reperfusion. So these are some of the conditions that leads to the production of free radicals. Now let's talk about the different classes or types of free radicals. We have reactive oxygen species such as ROS. Example of reactive oxygen species are superoxide ion, hydrogen peroxide, and hydroxyl ion. Reactive nitrogen species. Reactive nitrogen species are also examples of free radicals. Some drugs and chemical-derived free radicals are there, and also conditions favoring free radicals generation includes inflammation, some of the processes we've spoken about here. So knowing the definition of free radicals and the conditions that favor free radical production and the different types of free radicals are very high for the exam. The different types of free radicals are reactive oxygen species, reactive nitrogen species, and drugs and chemical-derived free radicals. And one thing for you to note here is that the hydroxyl radical is the most powerful free radical. That is a very common MCQ question in most of the university exams. What is the name of the most powerful free radical? Is called the hydroxyl radical. That is the OH minus. So now let's talk about how these free radicals are being produced. What are the ways in which some of these free radicals are generated? So we know that in every cell, small amount of reactive oxygen species form during normal mitochondrial respiration. So one of the ways in which free radicals are produced is during normal mitochondrial respiration, that is redox reactions of oxidative phosphorylation. In the presence of metals also such as iron, hydrogen peroxide is converted by Fenton reaction in highly reactive hydroxyl radical. So what we are trying to explain here, if those free radicals are produced, if one of the free radicals such as H2O2, hydrogen peroxide, it can lead to the production of another free radicals. How? If H2O2 reacts with Fe2 plus, another free radical is being produced, which is called hydroxyl radical. And this reaction is called the Fenton reaction. It's called the Fenton reaction. A popular MCQ question. What is the name of the reaction that happens when hydrogen peroxide reacts with Fe2 plus to produce OH minus? The name of the reaction is called Fenton reaction. Also, in the presence of superoxide is also another free radical. When superoxide reacts with H2O2, it converts the H2O2 to hydroxyl radical. So hydroxyl radical can be produced by when H2O2 reacts with Fe2 plus in the Fenton reaction, or when H2O2, that is hydrogen peroxide, is combined with superoxide. Superoxide is O2 minus. So when H2O2 reacts with O2 minus, it produces hydroxyl radical also. But the name of this reaction is called the Haber-Weiss reaction. It's called the Haber-Weiss reaction. So knowing these reactions are very important. The Haber-Weiss reaction, hydrogen peroxide reacts with superoxide radical to produce the OH hydroxyl radical. And the Fenton reaction is when the hydrogen peroxide reacts Fe2 plus and producing the OH radical. In the process, the Fe2 plus is converted to Fe3 plus. So the ferrous ion is converted to ferric ion. So knowing the name of these reactions are very, very important. Haber-Weiss reaction and Fenton reaction are all reactions that are used to generate free radicals. The Haber-Weiss reaction uses superoxide, while the Fenton reaction uses Fe2 plus. In order for you to know, it's simple. Fenton reaction starts with Fe, and Fe is the species that is used in the process of the Fenton reaction. Simple as that. So let's talk about some examples of free radicals or reactive oxygen species. As I said, superoxide is one of the free radicals. Hydrogen peroxide is another free radical, and hydroxyl radical. These are the most common free radical or the reactive oxygen species that we have. But it is also mentioning that, as I mentioned in the previous, the hydroxyl radical is the deadliest and the most powerful free radical. The hydroxyl is the deadliest and the most powerful free radical. These free radicals, when they are produced in our body, we know that they can cause damage to the cell organelles. But there are enzymes that are found inside our body that are responsible for the breaking down of these free radicals. Example of an enzyme that can break down free radical is catalase, superoxide dismutase, and glutathione peroxidase. High yield exam question. Name 3 enzymes that break down free radicals: catalase, superoxide dismutase, and glutathione peroxidase. So knowing some of these concepts will help you very well in your university exam. So now let's talk about the types of cell injury. What are the different types of cell injury? There are 2 main types of cell injury. They are the reversible cell injury and irreversible cell injury. Reversible injury may progress to a reversible stage and result in cell death. So reversible injury may progress to an irreversible state and result to cell death. So now let's talk about reversible cell injury. What is reversible cell injury? Reversible cell injury is a type of cell injury that happens if the stimulus is acute, brief, or mild. The cell injury produces change in the cells which are reversible up to a certain point. There are features that you see in a cell when the cell is undergoing a reversible injury. What are those features? Cell swelling is one of them. Formation of blebs. Blebs are elevations that you see on the surface of the cell membrane when the cell undergoes a reversible cell injury. Cell membrane is intact. On the reversible cell injury, the cell membrane is intact. The endoplasmic reticulum swells. The ribosomes detach from the endoplasmic reticulum. Autophagy of lysosomes. Lysosomes will start eating up its own organelles. That's what we call autophagy. And the last one is small mitochondrial. Then we have clumping of nuclear chromatin. So these are some of the microscopic features that you see in a cell undergoing reversible cell injury. And some of those features can be seen in the diagram here. The cell swells, the formation of blebs. Blebs are, as I said, are some of these elevations that you see on the surface of the cell membrane is intact. The cell membrane is intact. Endoplasmic reticulum will swell. Ribosomes will detach from the endoplasmic reticulum. The clumping of the nuclear chromatin. The nuclear chromatin will crump and the mitochondrial density will be very small. The mitochondria will appear fewer and small. And autophagy of the lysosome. The lysosome increase and digest its own cellular components. So these are some of the microscopic features that you see in a cell undergoing reversible cell injury. So now let's talk about irreversible cell injury. So if the cell is exposed to continuous injurious stimuli, or if the injury is severe, the cell. So obviously, irreversible injury happens when the stress continues to persist for a particular period of time, hence causing the reversible injury to transform to irreversible cell injury. And irreversible cell injury, as we said, it is irreversible. Only 2 things can happen, that is death, and the 2 types of cell death that can happen are necrosis and apoptosis. So that is basically irreversible cell injury. What are some of the features of irreversible cell injury? The features are: 1. The cell will shrink. Cell membrane is not in. Lysis of the lysosomes. Large mitochondrial density. Nucleus undergo pyknosis, karyolysis, karyorrhexis. Let's now talk about some of these features that we are talking about here. The cell shrinks. Here, as you see in the diagram, the cell size decreases due to loss of water. On the reversible cell injury, we see the cell swells, but here the cell shrinks. The second one we have is cell membrane is not. If you look at the cell membrane here, the cell membrane not in. The plasma membrane is damaged and loses its integrity. The integrity of the cell membrane is not in. And the third one is lysis lysosomes. Lysosomal membrane ruptures and hydrolytic enzymes are released into cytoplasm. So that is what we call lysis of lysosomes. Rupturing of the lysosomes, or the breakdown of the lysosomes, is what we call lysis of the. And the fourth one is large mitochondrial density. The mitochondrial density is large compared to reversible cell injury, where the mitochondrial density is small. And the fifth one is some of the nuclear changes that happens to a cell undergoing irreversible cell injury. We have pyknosis, karyorrhexis, and karyolysis. What is pyknosis? Pyknosis is when the nucleus shrinks and the chromatins are condensed in dense masses. The next one we have is called karyorrhexis. Karyorrhexis is when the nucleus breaks down into fragments, and karyolysis is when the nucleus dissolves and disappears. That is when the nucleus fades away. So that is the meaning of those three descriptions of the nucleus undergoing irreversible cell injury: Pyknosis, karyorrhexis, and karyolysis. Knowing these descriptions are very high yield for your university exam. So now let's talk about the differences between reversible cell injury and irreversible cell injury, which is asked in most of the exams. So let's look at this table. Let's start with the size. In reversible cell injury, we say the cell undergoes swelling. So cell swelling happens, whilst in irreversible cell injury, the cells undergo. Let's look at the membrane. So the cell membrane is intact in reversible cell injury, while in irreversible cell injury, loss of membrane integrity and the cell membrane is not intact. Formation of blebs. Blebs are just the elevations that are found on cell membrane. There is formation of blebs in reversible cell injury, whilst in irreversible cell injury, we don't have formation of blebs. Membrane disruption and leakage of cellular contents. Endoplasmic reticulum. The endoplasmic reticulum swells in reversible cell injury, whilst in irreversible cell injury, severe ER damage with fragmentation. The endoplasmic reticulum undergoes fragmentation. The ribosomes are detached from the endoplasmic reticulum, and here the ribosomal loss and cessation of protein synthesis takes place. Here the ribosomes are damaged or are detached from the ER. Here the ribosomes are lost and protein synthesis does not take place. Autophagy of lysosomes, and here lysosomes membrane rupture with release of hydrolytic enzymes. Mitochondrial, small mitochondrial density, and here we have large mitochondrial density in irreversible cell. Clumping of the nucleus, whilst in irreversible, the nucleus undergo three main stages: pyknosis, karyorrhexis, and karyolysis. Plasma membrane permeability. The plasma membrane permeability is no leakage of intracellular enzymes, while in irreversible cell injury, increased permeability with leakage of intracellular enzymes. And the last but not least of the factors that distinguish reversible cell injury and irreversible cell injury is reversible if the cause is removed. So meaning if the stress is removed, the injury is reversible. Whilst in irreversible cell injury, as the name implies, the cell undergoes necrosis, apoptosis, and the damage is. So knowing some of these different will be very important for most of the exams we are preparing for, and it will help us overall as people trying to study to understand the concept of cell. Thank you very much.